Welcome today to today's webinar and covering how tissues rewire in disease using deep learning. I'm Rick Young, a member here at Whitehead Institute, where my lab studies regulation of gene expression in health and disease. I'm excited to be your host and moderator today. Today's webinar is part of a series of talks that we've organized to share Whitehead Institute science and research with the wider community. In a few moments, we'll hear from our presenter, Whitehead Institute AI Fellow Na Sun. First, I would like to let you know that this webinar is being recorded for future viewing. Also, we will be answering questions at the end of today's presentation, but you can submit a question at any time using the Q&A box at the bottom of your screen. With that, I'd like to introduce our speaker today, Na Sun. Na began her scientific career at Lin University in China, where she earned a Bachelor of Science in Life Sciences. She then pursued a master's degree in bioinformatics and developmental biology at the University of Chinese Academy of Sciences, where she also worked as a research associate following graduation. In 2017, Ma moved to Cambridge to join the Raygon Institute of MGH, MIT, and Harvard as a bioinformatics specialist, contributing to computational analyses in autoimmune disease and immunology. In 2020, she began graduate studies in computational biology at MIT, earning a PhD in computer science in 2024. That same year, Na joined the Whitehead Institute as its inaugural AI Fellow, where she is developing machine learning approaches to study spatial and single-cell genomics in complex tissues and diseases. The Sun Lab at Whitehead Institute is developing deep learning tools to uncover how tissues recognize in response to genetic and environmental changes, while spatial transcriptomics has transformed our ability to map gene expression within intact tissues. Existing computational methods often overlook subtle spatial patterns associated with specific perturbations. To address this gap, the Sun Lab has created a new framework that integrates spatial analysis with contrastive analysis to detect condition-specific changes in tissue architecture. By disentangling meaningful biological signals from the background noise, this approach has revealed hidden spatial domains in both synthetic and real data sets, including those from disease and genetically altered tissues. Na will present this method and highlight how it can be used to uncover cellular adaptation and tissue remodeling, offering new insights into disease mechanisms, tissue plasticity, and potential therapeutic targets. Na, welcome and thank you.
Thank you, Rick. Hi, everyone. I'm going to share my screen. Hi, everyone. I'm very glad to share one of our ongoing efforts to uncover how tissues rewire in disease using deep learning techniques. So one of the biological questions we are very interested in the lab is to understand how do tissues respond to different types of the perturbations. And these perturbations could include the genetic mutations, environmental factors, disease conditions, or therapeutic interventions. And to understand the effect of these perturbations, there are multiple skills, biological skills, from genetic to molecular to cellular changes. And we are particularly interested in the intercellular changes, which is the spatial context, how the cellular how the cellular effect in terms of the perturbations. But before we dive into the how do we tackle these biological questions, I would like to show you some of the little games. So at the end of this book for the preschoolers, there's a small game which is to spot the differences between the two pictures. And the hint here is there are 6 to find. So basically, this is very obvious, and for most of us or the preschoolers, it's very easy to identify the 6 differences between these two pictures. And because this is pretty obvious visually, but if we look at these kind of complicated or crowded pictures, and there are more differences between the two pictures, it's a little bit hard to just identify those differences by naked eyes. But computational methods can solve this puzzle pretty easily. So if we transfer, or if we inverse the second picture into this panel, and we can combine this third image with the first image and do a little bit math, we can identify this picture with the grid and the non-grid parts. And actually the non-grid parts just represent to the 15 differences between these two pictures. And this is also computationally straightforward. However, if we just focusing on the biological differences in terms of the spatial genomics or spatial omics data. So for example, here, we do have the Whitehead brain tissues and also the Alzheimer's disease model brain tissues. It's not very easy or not very straightforward to identify the changes between these two brain sections by visually. And so because there are many different challenges, so those challenges may include the spatial misalignment from these two images, even though those two sections are from basically the same brain regions, but it's really hard to spatially align between each other. And another challenge is that the data is pretty high dimensional. So in this case, for each dot, it represents to a cell, and the colors is actually represent to different cell types. But for each dot, for each cell, it includes many genes expression, like hundreds or thousands. It depends on the how many genes are measured. But this just show this data is pretty high dimensional. And also across different brain sections for different spatial omics data is pretty high variation from the biological and the technical perspectives. More importantly, for most of the case, especially for the chronic disease, the condition-specific signals are very subtle, which means the effect is pretty small. So we have to develop very sensitive method to identify those small changes. And also, for each cell, the change is in the very complex tissue microenvironment. So try to evaluate the cellular changes, we have to consider the complicated tissue microenvironment effect. So that's why computationally it's not very easy, but there is a computational strategy, which is the contrastive analysis. This strategy is actually applied in the field of natural language processing. And the contrastive analysis is able to isolate the variations enriched in a targeted data set. For example, here, if we have a background, we have a target, and we can observe, we can see there's a specific signals enriched in the target data set. And through this contrastive analysis strategy, we can identify the signals which are only enriched in the target. So to use this strategy, we try to understand whether we can apply this contrastive analysis strategy in the spatial omics data. So to do this, the goal is try to identify the background domain, which reflects the tissue structures, and also the salient domain, which just highlight the condition-specific changes. So specifically, if we have a control section, we have a condition section. Through this strategy of the contrastive analysis, we try to identify this background domain, and this background domain is actually shared between the control and the condition. And usually, this background domain is actually representing the tissue structure or the architecture of the cell types, or the major regions. For example, in the brain, it could be different cortex or different brain regions. But in the salient domain, which the signal should be specifically enriched in the condition, samples, and this condition-specific changes is in the context of the spatial of the tissue. So to do this, we developed a spatial-aware contrastive analysis method for the spatial omics. And here, I would like to highlight that spatial omics include not only the spatial transcriptomics, but also the proteomics and metabolite measurements, if we do have these measurements in the spatial context. So in this case, if we have sections from the control and also the sections from the condition, through our method, we named it Haluka, we can identify two types of the embeddings for the cell embeddings for the condition section, for the condition samples. So the gray part represent to the background embeddings, and orange part represent to the condition specific or salient cell embeddings. And combined with the aggregation of the spatial neighbors, we could cluster those cells into different spatial domains. So with the salient cell embeddings, we can identify the spatial domains specifically in the condition. And also the background spatial domains are actually shared by the control and condition. So specifically, I would like to dive into the details of the method. And so here, these gray sections represent to the control sections, and these pink sections represent to the condition sections. And we are focusing on one spatial niche here. So here we have the gene expression vectors of this central cell. And with the neighbors, we have the covariation of the gene expression to represent the spatial neighborhood information of these central cells. And through this, we have these two types of the input data, basically. And we designed two encoders to take this input. And for the background representation encoders, which is shared by the control and the condition. But for the salient representation encoder, it only takes input from the condition samples. And through the reconstruction layer, we perform this self-supervised learning to reconstruct the gene expression and also the spatial the covariance of the spatial neighbors of the gene expression. To do after this, we can learn the embeddings for the background niche. And also the salient niche embeddings. So basically, for each cell from the condition, we will have two types of the embeddings, the cell embeddings. One is the background embeddings, and the other is the salient embeddings. So based on these cell embeddings, we can perform some downstream analysis for different datasets specifically. So to evaluate the performance of this method, we firstly generated the semi-synthetic data for benchmarking. And the real data or the original data is actually coming from this study, or this is morphish from the mouse hypothalamus, and is measured about 155 genes. So here just show one example of the brain sections colored by the cell types. Again, this for in this map for each dot representing one cell, the color represents for different cell types spatially. And this is original data, so we can treat this data as a control. And to generate to generate this synthetic data as a condition, we sampled, we draw the values by sampling from the gamma distribution based on the combinations of these two parameters of the gamma distribution. And also we use another parameters, which is the size, to decide how much of the brain regions could be covered for this signal. So for example, here we have three sides of this signal, and the values for each dot actually follow the gamma distribution with these two parameters sampling. And on top of this original data, we added these values into this original data, and this combination could be treated as the condition. So with this control and condition, we performed the benchmarking analysis. So in this case, the background, the ground truth, actually, we use the brain regions as a background. The brain regions information are coming from the original paper, and the salient domain we identified as a ground truth is actually the signal we added, we simulated in the data. So our baseline methods here include contrastive VI, which is a single cell contrastive analysis method designed for the single cell gene expression only, without consideration of the spatial information. And another baseline method is the cell charter, which is a spatial clustering method or spatial domain identification method. We also include a method called cinema OT, which is a causal independent effect module identification with optimal transporter. And to compare with these three baseline methods, we found Haluka, our method, could outperform in salient domain identification. So in this panel, we can see Haluka actually could very well capitulate the signals we simulated. And also actually it can identify the background domain pretty well compared to other baseline methods. But this is just one example to show one data set we generated. And quantitatively, we use two different metrics to evaluate the performance, which are adjusted rank index and also the normalized mutual information. So from these quantitative results, we can see Haluka can outperform in this salient domain identification significantly better than other methods in different simulated datasets. And also we can see from the background domain identification, Haluka could also perform well compared to other methods and very stable. The results are very stable. So this just suggests the power of the Haluka, the spatial aware contrastive analysis method we developed are able to identify the informative information in the condition specific samples. So we applied this method in many biological datasets here and try to indicate the general applications of this method. We include different types of the spatial transcriptomics techniques, such as the star map, Morphish, 10x Xenium, and also we include different modalities. For all of this, they are spatial transcriptomics, but we also have the Cordex data, which is proteomics. It depends on the panel. So in this case, we may have 58 marker genes, antibodies. And also we indicate we show that our method can be applied into the multimodal data, like the combination of the RNA and the protein. So we also, in our case studies here, we also include different or multiple biological contexts, such as the mouse AD mouse model, the disease mouse model, and also the exercise in the normal aging in mouse brain. and also in some of the human lung disease and cancer context, and also the genetic perturbation studies. So this just suggests the general or the broad applications of our method. So today I will show you some two of these case studies to show how do we use the background domain and the salient domain we identified for the downstream analysis to provide the biological insight. So the first example I would like to show is the application in the human lung fibrosis 10x xenium data. So this paper published earlier this year in Nature Genetics, and in this study, they measured spatial transcriptomics in human lung fibrosis in 3 different pathological groups. It includes the control group, the less and more affected lung samples. And the panel here they used is about 300 gene panel in 10x xenium at the subcellular resolution. So in our case, we treated the healthy or the control samples as the control in our Haluka framework, and the less and more affected samples as the condition. So with this background domain and the salient domain, we performed the downstream analysis. So I will just show you some of the examples. This is the original transcript niches and cell niches identified in the original study. And in our method, we identified the background domain and the salient domain, and we showed the enrichment of the background domain is actually significantly enriched in some specific transcript niches and also the cell niches. So here, this heat map show the enrichment of the background domain in the transcript niches. We can see the specificity of the enrichment, which just suggests the background represent to the overall tissue architecture or tissue structure. So with this background domain, we can also apply this background domain representation to do the spatial alignment. So the spatial alignment here means if we have 2 different spatial samples, we try to align or registrate the spatial coordinates from 2 separated tissues or sections. So here we evaluate whether the background domain representation we identified by Haluka is actually better than the original gene expression only driven spatial alignment. And we do observe a little bit better performance of the spatial alignment between different samples with background domain representations. And with this, we applied this background domain representation driven spatial alignment techniques into the samples from this human lung fibrosis dataset. And we have the less and more affected samples. And we just show here that in the early stage or in the less affected lung tissues, the homeostatic fibroblast actually is already priming for the later pathological response of the fibrosis. So here we can see some of the cells or some of the fibroblasts actually can transit into the inflammatory fibroblast or activated fibroblast in the case of the more affected pathology of the lung fibrosis. So which suggests the potential application of the background domain for this spatial alignment to align different stages of the disease samples. So next, we try to evaluate the applications or the biological functions of the salient domain representations we identified in this dataset. And actually, we found the salient domain representations can distinguish the samples or the sections by degree of the effectiveness, which means we performed this PCA analysis. We found PC1 can distinguish the more affected and less affected pretty well, significantly. And in contrast, if we use the gene expression driven PCs, we can see actually the PC1, the PC2 are really bad to distinguish the samples from affected, less affected, and more affected samples. So which suggests the salient domain representations of the cells reflect the real pathological related changes, which could be hidden in the raw gene expression. So this is our first applications in the human lung fibrosis. And also the next example I would like to share today is the immunotherapy on melanoma with the cortex proteomics data. So published like 2 years ago, this paper performed the cortex multiplex imaging to show the 58 proteins expression at the single cell resolution before and after the immune checkpoint inhibitor. And in total, they include 6 patients, and 3 of them are responders, which means those patients are responding to the immunotherapy, and 3 patients are non-responders. So in this case, we applied our Haluka and we used the pre-treated individuals as a control and the post checkpoint inhibitor treated sections as a condition, so that we can identify the background and salient domain representations for each cell for all of these 6 patients sections. So and also here I'm showing you one example of the responders sections, and here we have the visualization by the cell types and the neighborhood, which are provided by the original paper. And in our analysis, we identified a group of the background domains and also 4 different salient domains. And we can see actually the background domain are really related to the cell types and the neighborhood. But the salient domain is kind of interesting because if we use the salient domain representations to perform PCA as what we did in the human lung study, and we found the PC1 and PC2 can actually distinguish the responders from non-responders pretty well. And then we evaluate what's the distribution or what's the proportion of the salient domain in the responders and non-responders. So here, this bar plot will show the proportions of these 4 salient domains, and 0, this first column represent to the non-responders, and this on the right, the panel is the bar is the proportion of the 4 salient domains in the responders. And from this analysis, we can observe that the salient domain 1 and 3 are actually significantly enriched in the non-responders. So that we wonder what's the cell types enriched in these domains. So we plot this proportion of the cell type categories, and we found, interestingly, in salient domain 1 and 3, the number of the immune cells are significantly reduced, which basically explain why there's no response to this immune checkpoint inhibitor therapy. And even though those domain 1 and 3 are significant in the non-responders, and their immune cell type proportion are also pretty different, which means especially if they form different spatial domain with the tumor or the stromal cells, the immune cells in the spatial niche are pretty different. So this may also suggest that if we focus on this specific domain, we can identify more different, more like divergent tissue spatial architectures of different cell types, providing some biological insights to understand why these patients or individuals are not responding to the immunotherapy. So furthermore, we focused on the domain 3, which here we use the domain 3 cell embeddings as a reference to integrate with the single cell RNA-seq data. For the single cell RNA-seq data, there's no spatial information. So with the MaxFuse, we do this integration of the proteomics and the single cell RNA-seq, try to map the single cells from the single cell data to the proteomics data, so that the assumption here is that if the cell from the single cell RNA-seq data can align or can match with the domain with the cells from the cortex data in domain 3, those cells may not be able to respond to the immunotherapy. So with this, we can have two groups of the cells or two subpopulations of the cells in the single cell RNA-seq, which we cannot get that information only from the single cell RNA-seq data only. So that we can compare whether we can identify some molecular mechanisms to drive this bad immunotherapy response. So here, this UMAP just showed the integration results of this public single cell RNA-seq data and also the proteomics data, the cortex data we have here. And with this, we identified differentially expressed genes between cells in response and non-response. Specifically in these CD8 T cells, we identified a group of genes which are upregulated in the non-response cells and downregulated in non-response cells as well. And gene ontology enrichment analysis showed these genes are enriched in the regulation of cell migration, cellular response to interferon gamma and antigen processing and presentation related. So for the downregulated genes, they are kind of related to the protein targeting. So overall, these dysregulated genes and their functions suggest the potential inflammatory suspension to the poor immune therapy outcomes. So this again, the salient domain we identified in the condition sections provide some biological insights to understand the molecular mechanisms of the immunotherapy response. So to summarize, in our recent work, we developed Haluka, which is a spatially aware contrastive analysis method, and to identify the condition-specific spatial domains. And in our simulation studies, we found Haluka outperforms both single-cell and spatial clustering approaches, achieving state-of-the-art performance. And it also demonstrates the strong robustness and the general or the broad applications in different datasets across different spatial transcriptomics techniques and also different biological contexts. And more importantly, we show that Haluka's background and salient domain we identified enable the powerful downstream analysis extending its utility beyond domain identification, and also providing the biological insights which we can't identify it based on the regular or traditional single cell spatial transcriptomics data analysis. So with this, I would like to take this opportunity to thank our previous research intern, Yan, who is leading this project. Yan is doing his PhD program now at University of Pennsylvania. And also I would like to thank my lab members, Sophia, Albert, Yuxiang, and Sumiko for their support with this project and the lab. And also I would like to thank Andy and Craig from Whitehead IT department for their computational support and discussion about the computational resource in the Institute. And I would like to thank my faculty mentors from Whitehead and also MIT ESS, including Ruth, Rick, David, Jonathan, Polina, and Colin. And also thanks to the human resource and the Whitehead facility for their support to my lab, and also Whitehead Innovation Initiative for their support to this project and many other projects in the lab and the lab overall. So thank you for your attention, and I'm happy to take any questions.
Thank you very much, Na. I'll relate some of the questions we have for you from the audience. If I could just start, I'm wondering, your technology really highlights how complex certain systems are. For example, you picked the immune response and how that response changes with therapy. You're dealing with many different cell types in that immune response that you're profiling and analyzing. Is there a limitation to the number of data sets that you use? How does that scale as you use more and more data sets?
Thank you, Rick, for this very good question. So actually, our method here I've just presented is not very sensitive to the scale of the data. If you do have like for example, two biological replicates as a control and two biological replicates as a condition, that's pretty good to be used for this method. And also, if we do have a bunch of the controls, a bunch of the conditions, that also work. It just depends on how much of the computational resource we have. So actually, the contrastive analysis or this variational autoencoder based structure we have here is pretty computational time saving. So the cost is pretty small actually.
Mm-hmm. Do your computational approaches capture dynamic transitions, or are you looking more at static snapshots of cell states, or can you do both?
Actually, we can do both. So in the case, when we do the setup of the experiments, of the computational experiments, we do have the control label and the condition label. But in the condition, we could have multiple time points. For example, in our human lung fibrosis data, we do have a less affected samples and also more affected samples, but we treat them all as a condition. But eventually, the salient domain was identified. The representation can still distinguish these two groups of the pathological samples, which means the salient domain can capture the differences between the samples. And in another experiment, actually, we do have 4 time points. We have a control, and also we have onset, peak, and remission. And still, we can identify some salient domain which are specifically in one stage, which means the method can capture the dynamics of the time course data as well.
How transferable are the models you're using across tissues types, diseases, or even species?
So the setup here or assumption here is that as long as you have a control and condition, it's very data set independent. So we only do the self supervised learning only inside of the data. So we didn't transfer the information from one data set to another.
Have you uncovered any surprising patterns of tissue rewiring that challenge existing biological models?
That's actually very good questions. We don't observe very weird, but we do observe some like very small spatial niche, which could be learned as an independent salient domain. And this pretty small spatial niche, they do have very sensitive to the condition. So I think it's pretty nice to have this method, which is sensitive to the very small effect, and in the spatial context.
Given the complexity and high dimensionality of biological data, do you need to take steps to reduce overfitting?
That's a very interesting question. Yeah, so I think the overfitting mostly um, could be coming from the low number of the replicates. So especially for the human samples, for example, we have to consider the variations from the technical and also from the biological. So based on my experience, based on our experience, I think for the mouse case is pretty the variation in the biological replicates or technical replicates are pretty small. But if we use the human samples, for example, the AD samples or the tumor samples, they always have some like variation, which are specifically to any kind of potential reasons, like environmental factors or even genetic factors. So if we do have very small, or we only have one or two biological replicates for the control and condition, sometimes we do have some overfitting, which means the signal we identified may not be reflecting the real disease related, but maybe could relate it to some confounding factors. So that's basically the major issue. Yeah.
Yeah, and that challenge is not specific to Haruka. It's a general issue that we have in analyzing large data sets.
Yeah, I think so. So basically, that is actually the general issues for most of the single cell data analysis and for the spatial analysis. For the spatial analysis, we do have like more confounding factors, like whether the regions we captured are really kind of mostly matched between samples, between conditions. And that is I think that is another confounding factors which are not basically from the single cell data analysis, but we have to always aware those kind of confounding factors and try to see whether the information we identified are really, I mean, biological relevant based on the purpose of the analysis.
And I presume that you see opportunities to combine these types of deep learning approaches with lab-based perturbation, like CRISPR-based perturbations, to test hypotheses about tissue rewiring. Is that right?
Yes. So when I showed the like multiple applications in different data sets, we do use the genetic perturbation spatial readout. That data is from Jonathan's lab. So basically in the liver, they performed about 200 genes perturbation in hepatocytes. And we try to apply our method to identify specific salient domains, which include or enriched some of the cell types. We do observe the spatial proximity change of the some subset of the hepatocytes and immune cells or vascular cells. But if we want to do more analysis, we have to just dive into that specific domain we identified and also the specific genetic perturbation. So that is also one basically potential follow-up study to just specialize this Haluka method or contrastive analysis in the single gene perturbation and output genetic perturbation to distinguish or to see which factors are actually from the genetic perturbation or from their spatial neighborhood. So yeah, I think which is kind of different from the regular control and condition, because the condition is actually the combination of different genetic perturbations with the spatial neighborhood or spatial context. So we would like to do more to see whether we have a better computational framework to do that. But currently, the application in the genetic perturbation data is that with this genetic perturbation overall, we can identify some specific spatial domain, which may be enriched in some of the perturbations of some genes. And those genes could be functionally relevant. All the downstream gene expression they introduced are shared with each other. So that's the overall patterns of the genetic perturbation. But if we try to do more or in-depth analysis on the effects of the single gene genetic perturbation or the combinations of the genetic perturbation and the spatial context, we have to develop some very specific method for that.
And there's a technical question. How was Haruka performance evaluated and what metrics were used?
So the question is about the evaluation metrics, right?
Yeah.
So the evaluation metrics here we used is AII and also normalized mutual information. So which are metrics to evaluate how two groups of the clustering or the two groups of the clusters are aligned with each other. So basically for our ground truth, we do have the, for example, in the semi-synthetic data, we do have the 3 or 4 different salient domains. We know that is the region we give the simulated signals. And in the background, we do know that is the predefined Bruin regions. So for each cell, we know their label. Their label would be the names of the clusters or the names of the Bruin regions. So with this, we could do this comparison with this ground truth and the prediction from Haluka and many other methods. So with these 2 vectors of the labels or groups, we can use the evaluation metrics we used in this study to see how well. So yeah, so basically those are well-known evaluation metrics for the cluster classification comparison.
Yeah. What kinds of biological problems would you like to next explore with Haruka?
So I think in our current experiments, we do observe some applications of the salient domain and also the background domain. For example, in the human lung fibrosis data, we found the potential applications of the the background domain representation could be applied for the spatial alignment. So for example, in most of the cases, if we have the control spatial sections for the spatial transcriptomics, and in another diseased samples, we cannot see, oh, these spatial sections are exactly from the same section, because they are not, actually they are not. But if we try to identify some some disease-related signals, we may want to align them together. Or so from our study, we do see the potential of the background domain could do this spatial alignment pretty well. So we would like to do more on that. And from the salient domain perspective, we do observe the some of the salient domain, they are very relevant to the disease. So they do enrich, they do have some enrichment of the specific cell types. And we will dive into that small spatial niche to see which ligand receptors or which signaling pathways are actually contributing to the cell-cell communications in that small niche. And if we, for example, if we consider CD8 T cells, if these CD8 T cells belong to this salient domain, and we show this domain is related to the immunotherapy, for example, and so we can just focusing on those subpopulation of CD8 T cells rather than all of the CD8 T cells, so that we can specifically evaluate how these CD8 T cells interacting with other cells as neighbors, and then we may identify some of the ligand receptor candidates which could drive this disease-related response. So I think the salient domain provides us higher resolution of the spatial the cells in the spatial context, so that we can just focusing on those small populations rather than all of the cells based on their gene expression identification, so that we may be able to identify some new targets or potential candidates of the therapy. So I think that's basically the application of the background domain and also the salient domain representations can tell us.
Aruka does some really amazing things with these data sets. And I'm wondering, do you see it standing alone, or do you see it at some point in the future, this becomes an agent within a multi-agent apparatus?
That's a nice question. So I think it is basically it's not standalone. I think in the future it depends on the like the AI agent or those kind of functions. We can basically try to combine this this output of the Haluka and with AI agent. For example, one of our current efforts is try to interpret the salient domain we identified. So sometimes we only have the cell proportions in that domain, but whether this domain is really actually representing to a biological related unit. So we try to integrate with the prior knowledge, and so that we can use AI agent to see whether we have a better interpretation of those domain we identified. I think that's one potential combination with the ongoing, the AI or large language model driven agent and techniques.
Well, Na, I want to thank you for a very informative talk, and I learned a lot. And I want to thank our audience for being here and participating.